Okay, we're back. And this, thank you. This topic is a little bit more complex, so I did want to give it its own video. Last time we talked about the central dogma. DNA can get copied in a process called replication. We can make it's not an exact copy of DNA. We can kind of take a little piece of DNA, copy a recipe from DNA's giant cookbook, and take that out of the nucleus, and we call that transcription. And then we can turn that recipe into a product, a protein, and we call that process translation. So let's just take a look and go through, and we're going to go ahead and look at these processes, transcription and translation. Oh, and proteins make your traits, like your hair color, or your eye color, or your skin color, or your height, or other things. If you're a flower, your flower color. So there. So let's talk about the real job of DNA. It codes for your traits. It makes you. It's the instructions on how to build, in your case of your DNA, it's the instructions on how to build you. If it's a cactus, it's the instructions on how to build a cactus. Oh, I gotta be careful. That's right, this thing was leaking battery acid. Oops. Nothing happened. Everything's fine. So the instructions that are found in DNA get turned into a protein by the ribosome, and we need to talk about how this whole process happens, okay? Because there's a lot going on. We're going to kind of summarize this process. We can go way deeper into it, but for right now we're just going to try to summarize it. So transcription is when a section of DNA, a gene, is turned into something called mRNA, messenger RNA. That's what the M stands for. So why does this happen? Well, DNA is humongous. Okay, it's this huge molecule. We have 23 pairs of chromosomes. We've got hundreds of different genes, probably thousands of different genes that say, here's how you make this part of a person. It's very important. It's like we don't want this DNA to get ruined. If DNA has lots of mutations in it, well, that could cause cancer. So we don't want DNA to be damaged or changed. We want it to be nice and safe. But with messenger RNA, it's like we can take a small copy of DNA and it can get sent out to the world. And if it gets like wrecked up, who cares? It's okay. So I've tried to say this before. I'll try to give you guys an analogy. DNA is like Grandma's cookbook. Like Grandma gave you her cookbook that she cooked all her like best recipes with. And she gave it to you. Like me. It's very, very old. Okay, your DNA is not that old. It's as old as you are. But anyway, well, we won't go there. That's another discussion. But anyway, it's something that's precious. So if like your DNA and like this cookbook. So if I wanted to make one of these recipes that she made, I don't want to take this like very old, like 70-year-old cookbook out and like just throw it in the kitchen where I will wreck it up, or my lovely child will get flour and oil on it, and then it's ruined. But I can go and I can take a piece of paper and a pen and I can jot down the recipe I want. And if that gets wrecked up in the kitchen, well, who cares? Like, it's probably going to go in the trash anyway. No big deal. Like, it can get thrown away. Messenger RNA is like the copy of the recipe I made from the cookbook. DNA is like the cookbook that I don't want to get damaged. But messenger RNA, it's okay. It's small. I don't have to carry this big, heavy cookbook around and lug it around and like find the page 50 times because I keep losing it or because it keeps folding closed or whatever. Nope, I just copied one gene, one recipe. I took it to the ribosome, the kitchen, so that way I can use it there. So that process of me copying one little recipe, copying one gene, it's called transcription. Now, we don't copy to more DNA. We copy to the special messenger called messenger RNA. So let's kind of look at that. What's that thing like? Well, it kind of almost looks like half of a DNA. It's single-stranded, not like double-stranded like DNA. Instead of having thymine, T, it's got a pinch hitter in here, this U, uracil. Why does that happen? Thymine can't bond to the sugar ribose in RNA. So you just need to know thymine can't live there. Okay, it's not good there. But you can think of uracil as like, oh, it's like this, okay? Like thiamine's the quarterback, and the quarterback's like injured, or they're like, let's give the rookie a chance, okay? So we have someone substituting in. They do the same job. Like if we have the backup quarterback, they're still a quarterback. They're doing the same job. They're just a different person, okay? It's well, Josh Allen. Who's the backup for Josh Allen? I don't know. We'll pretend like it's Tyrod Taylor, even though he doesn't play for the Bills anymore. Okay, I know it's wrong, but still. Okay, they're both quarterbacks. We just subbed one in for another. And it's much smaller than DNA. Okay, so this is like a small section. Okay, it's not even close to being as big as DNA is. So how does it get made? How do we do it? Is there like a person in there copying this messenger RNA down? Nope. In fact, transcription happens a lot like replication. DNA is unzipped, an enzyme comes in, and this is, instead of DNA polymerase matching DNA, this enzyme is called RNA polymerase, and it matches RNA bases and it matches them up. So C and G match, G and C match, T will go to an A, and then if there's an A in DNA, remember, RNA does not have that base thymine, so A will match with U. So here's a life pro tip. If you're taking a test and they give you a section and then it's got like A, U, G, C, C, A, and they're like, is this DNA or RNA? It is RNA because it has U in it. So it's kind of like a tip off. Or if they give you a section of DNA and say which one of these would match if we were making RNA, you have to make sure it has the letter U in it. If it has a T in it, that's wrong. So I have a little thing for you to remember. A will match with U, C to G, and T in the DNA will match with A. And then when this messenger RNA is complete, it leaves the nucleus. So just like once I've copied my recipe down, I leave the back room where the cookbook is and I go to the kitchen. So I'm going to go where I'm going to make the food. So the messenger RNA is going to go where the protein gets made. So you guys can always take a look at this difference between replication and transcription. So let me kind of talk to you about this process of translation. And I'll try to make sure you guys understand the significance of it. All living things are similar in how they're made up, like the chemistry that makes them up. The nutrients are the same. And then all living things are made up of some kind of cells that have DNA inside them, which is the blueprint to make living things. So depending on what DNA it has, what blueprint, we get a different living thing. Like a kitty. So we get all the different life we see on Earth from this limited amount of products, or this limited amount of ingredients. So let's figure out how that happens. How can we get all different life on Earth, including just all the different humans we see? Not even thinking about all the different animals and plants and fungi and protists and bacteria and archaea and stuff we haven't discovered yet. So we have raw materials. We'll think about it like building things. We have a blueprint. We have tools. We can make something. Okay. Well, our bodies have raw materials. We call those nutrients. It's like the food we take in. We have a blueprint. That's the DNA. We have tools. They're enzymes that build or break. And the product for living things is like the dog instead of the doghouse. Well, just like if we have different blueprints, we could have the same materials, same tools. But if we have different blueprints, we can make a doghouse, or maybe we can make a table and chairs. Well, with the same nutrients, we have different DNA blueprints. Oh man, I should have switched the human and the dog, and then my head would have been in the right spot for once. But with different blueprints, even with the same tools, the same enzymes, the same nutrients, we get different products. And the reason for that is this transcription and translation, reading what the DNA code says. So that's why the blueprint's so important. So let's talk about what translation is. Translation is the process of messenger RNA going to a ribosome and being turned into a protein. So this is kind of like a complex process. I wish we had been here in person because it's much easier when we're doing some of the work for this in person. But here we are. So hopefully this will help. So how does this happen? Well, get ready. Messenger RNA leaves the nucleus. It's going off to college. Not really. So it's like me leaving the back room with my recipe. And it goes to the ribosome. You can think of the ribosome like the kitchen. Ribosome is an organelle that makes protein. So the ribosome will read the messenger RNA. It reads it 3 bases at a time, and those 3 bases are called a codon. So actually the messenger RNA goes into this ribosome kind of like a dollar bill going into like a candy machine or a pop machine. And that reads the dollar bill and it's like, yeah, it's probably $1. Here you go. Here's your candy. Well, the ribosome reads the messenger RNA in a very specific way, and it reads it 3 letters at a time. So you can imagine to the ribosome, each of these 3 letters is sort of like a word that it has to match up. So now we know where the messenger RNA went. In transcription, we saw messenger RNA got made. Now we know where it goes. It goes to this ribosome place. The ribosome is going to read this messenger RNA, look at the 3 letters, and grab this weird key-shaped thing, this golden thing, which is called a tRNA. T stands for transfer. Wouldn't it be awesome if it was like translation RNA, because it happens in translation? It's not. It's transfer. Okay. Sure, scientists, it's fine. You name things whatever you want. We'll just learn them. Okay, so the ribosome reads the 3 letters on the messenger RNA and grabs up a tRNA and grabs it up and like matches the letters. Remember how we matched letters for replication and for transcription? Well, the letters are going to be matched again. So A and U, G and C, U and A. It's going to read the letters and match them. So whatever's on the tRNA, that's called an anticodon. So here's our codon down here, and here's our anticodon up here. So it's illustrated, it's really written out for you. This tRNA attached onto here, so this little bead here, that's an amino acid. So that's why it's called transfer RNA, because it's going to transfer an amino acid in the ribosome. So like I said, the tRNA has an amino acid attached to it. So the ribosome's job is this: read the messenger RNA, grab up the matching tRNA. Then it's going to connect the amino acid that transfer RNA has. It's going to connect them together like connecting little beads on a string. So it reads the recipe in order, that messenger RNA recipe, and it takes the ingredients. It's really like cooking in a way. It takes the ingredients, we call those tRNA, and it says, okay, well, I need one of these, and now this one, and now that one, and puts them together in order. And then at the end, when they're all connected, that makes a protein. That those amino acids will connect, fold around each other, and make a finalized protein. And that's how you have things like skin color, because melanin is a protein. And your DNA was like, okay, we've got to make some of that. Get on it. Yes, sir. Yes, ma'am. Okay, and that's translation. So this is, even though my head's blocking, this is like the Rosetta Stone of DNA language. So scientists figured out what messenger RNA code is going to match with what amino acid. So now, not only can we read all the DNA code, we can translate for us that code to say what amino acids are we making, so we know the ingredients of every protein. We could. I'm not sure if we do know this yet. We can know the ingredients of every protein that make us up. So, for example, if we look here, AGU. All I have to do is use this chart. Okay, here's our first position, A. So everything in this row is going to start with A. Okay, that's kind of hard. There's a lot of things. I'm looking for AGU. I don't see it right away. I'm scared. Well, okay, calm down. It's okay. We look up here for our second position, G. So everything here in this column, the second letter is G. So if we match them up, AG, here we have AGU. That gives us this serine. So if I have AGU, that means the amino acid that was on here, it happens to be this green one, was serine. Okay, so we understand exactly what codes for what. So if you're looking at this, and I will bet you that this will be on your final exam. If you have a final exam in biology, which you do, it's going to be on there. You're going to need to know how to read this. And it's not bad, I promise. They'll give you some letters. They'll say which amino acids match up with this RNA code. Well, you have to look at them 3 letters at a time, and then just look at the boxes. Always start here, okay? And the whole row, that's the letter U. Everything starts with here. So everything here starts with the letter U. Okay? Easy. I promise it's easy. So let's just take a look at one last thing as we go from a gene to protein. And that'll be like transcription and translation. So we've got our very valuable DNA here in the safe, in the nucleus. We don't want that to leave. That's kind of important. It's the master blueprint. Well, if I'm going to build anything, I need to look at the blueprint. Okay, well, we're not getting no concrete on this main blueprint. Make some copies. All right. We call that messenger RNA. That's some copies of this blueprint, or at least part of it. Maybe I don't need the whole thing. I'm not working on the 5th floor. I'm only on the 1st floor. So I'll make some copies of that I can use at the work site so I don't wreck up the original. We call that transcription. Messenger RNA is going to leave the nucleus, by office, and it's going to go to the work site. It's going to go to the ribosome. There it is. And messenger RNA's job is to make whatever's on this blueprint. And the ribosome is the factory that's going to do that. Okay, and it's going to make protein. So here's our ribosome protein factory. Well, the ribosome is going to grab up these tRNAs. So it's coming here as a delivery truck. It's delivering amino acids. So in the ribosome, it's receiving the tRNA and the amino acid, and it's putting them together in the correct order, according to the DNA blueprint. And so there it is there. It's put together in the right order. It's made a protein. It's behind my head, which makes a trait. Oh, sorry. That process, that's translation, which then will make a trait, which makes you. So that's transcription and translation. So if you can look at this picture and kind of tell the story of what's going on, you're good. I would say right now, pause this video. I'll take a drink while you're pausing. Water's good and good for you. Oh, I should have told you what you're going to do when you pause. Anyway, pause the video. Take a look at this picture. See if you can name what's going on or describe what's going on in this picture. And if you can, good, you've got it. You've got transcription and translation. If you can't, take a look at some other resources. There's so many good videos about this. If mine doesn't help you that much, really search DNA transcription and translation for students or for kids. They'll have stuff for you, I promise. So pause this video, and then I'll kind of give you like some of the things that are here, or tell you what some of the things are. Did you pause the video? What are you waiting for? Pause the video. Come on. Try it. Seriously, try it. All right. So I'll assume you paused, or you're just not going to. And I'll tell you what these things are. So we've got DNA. We've got helicase, unzipping. And you can think of this as all the enzymes too, I guess, making your messenger RNA, so RNA polymerase. So this little ribbon here is RNA. Look, whoop, it went right out of this nucleus. This little brown thing is the ribosome. I know they look like little cans here, but these are the tRNA. And these little geometric shapes here, like this hexagon here, those are different amino acids. So now that you kind of know what those are, look at that and see if you can describe what's going on in this picture. And really, it's what we just talked about. So I hope that helps, hopefully, and I will see you later. Bye.